Hello all, my name is Maleesha Marakkala. I am from Department of Chemistry and Biochemistry. I am going to present my research, Interactions of CHIP and AMPK protein complex. Proteins are small molecules that ranges from 1 nanometer to 100 nanometer. So it is pretty challenging to visualize interactions between two proteins, especially when we need to know which part of the protein interacts with the other protein. In my research, I am studying about interactions of two proteins called CHIP and AMPK. This is my overview. I am going to introduce the structure and function of CHIP and AMPK proteins, and then hypothesis and experimental design. After that, results. Let's look at CHIP protein. That is a long term is carboxyl terminus of HSP70 interacting protein. It is a multifunctional protein. CHIP contains three main domains: uh, tetracyclopeptide repeat, U-box domain, and TPR coil domain. Among these uh, uh, domains, um, they have many different functions. So among these functions, acting as an autonomous chaperone for adenosine monophosphate activated protein kinase is the interested side in my research. Uh, Adenosine monophosphate activated protein kinase protein is a heterotrimeric protein. It contains three subunits: alpha, beta, and gamma. Each of these subunits takes an specific role in both stability and activity of AMPK. The alpha subunit may exist as two different isoforms called alpha 1 and alpha 2. These alpha subunits contain a specific domain named as autoinhibitory domain. Uh, Preliminary studies shows that uh, this domain, AID domain, is interacting with chip protein. Mm, my hypothesis is chip increases AMPK activity through direct interactions between chip and the AID. Uh, so from preliminary studies, we know that the interaction is going in between chip and the AID. We just do not know which domain of chip is involved in the binding. So my aim is to determine the interactions between chip and AMPK, and the second aim is determine what is the domain of chip protein that interacts with the AMPK AID. This is my experimental design. I am going to purify these protein parts first, like U-box domain separately, CC and TPR domains separately, and all together. That means all blend protein, chip protein, and for AMPK AID domains, alpha 1, alpha 2 domains, alpha 2 AID domains separately. And then finally, I will perform biolayer interferometry to determine binding constants. Let's look at BLI. What is BLI technique? Biolayer interferometry. It is an optical analytical technique. It can analyze the interference pattern of white light reflected from a layer of immobilized protein on the biosensor tip. As indicated in the image, first we can add our first protein. That means I will add chip here, and then um, it will bind to the biosensor part of the machine. So this thickness of the biolayer has increased now. After that, we can add the second proteins, the AID domain here. So if the second protein binds to the first one, the whole thickness of the biolayer again increases. Uh, then because of this change in the thickness, the reflected white light pattern also changes. Therefore, we can get the idea whether the proteins are interacting each other or not. In my results, I got protein expression. I did protein expression and protein purification for some proteins. I started purification of chip full length protein, and I finished purification of AMPK alpha 1 AID domain. Uh, Once I finish the chip protein purification, I will do bivalent interferometric assays, and then if the proteins are interacting with each other, I will get at least 10 to the power minus 8 binding constant. Mm, in order to do BLI assays, we always need good purity of proteins. So I purified the AMPK alpha 1 AID protein, and I visualize it in a protein visualization method called gel electrophoresis. In the first lane, it shows a protein marker which gives non-size proteins, and um, from that we can get the idea what is the size of our protein. So I purified my correct protein as showing in the image. That means it is in between 37 and 25 kilodalton. So my protein is 33 kilodalton. So I am sure this protein is purified now. And further, I run the second gel, shows the concentrated form of the protein. So that indicates my protein is not contaminated with even a small amount of other proteins. That's all for now. I would like to thank to my Page Lab members and Dr. Page and Dr. Jonathan Schuyler, and thanks to you all. These are my references. Thank you.